I'm really going to continue along the biological venue. I'm going to talk about why the microbiome that forms early in life is important and what happens when we change it. And the context I want to begin with is something that all of you know, is that in recent decades, we've had a number of diseases rise. A few of them are shown here. There are many more. One disease in particular that I want to talk about is obesity. I think most of you are familiar with maps like this. This is from the CDC. And what it shows that in 1989, there's no state in the United States where more than 14% of adults are obese. But by 2010, there's no state with less than 20% obesity, and the national average is about 30%. What's remarkable is that it is happening all over the United States, and the distance between the first and the last map is only 21 years. Something very powerful must be going on. It's not just in the United States. It's all over the developed world. Here's data from the Netherlands over about a 30-year period. You can see how the map is getting bluer. And this slide looks at obesity trends among US children and adults and adolescents. This is from NHANES data over about a 35-year period, looking at different ages in childhood, and we can see the way the trend lines are going. So even in the youngest group, obesity is rising, and this is representative of a wide body of data that the roots of adult obesity begin in childhood, in early childhood. Now, it's not just in developed countries. There's a big problem in developing countries. This is from public radio, just to announce the problem. 10 million children in Africa are now obese. This slide, which may be difficult to see from the back, looks at rates of overweight and obese children globally by level of development. This is in children under 5. And I apologize, it's so small, but it's just because I made it on Saturday. And the rate in developed countries the prevalence is 12.9%, and in developing countries it's 7.2%. So it's clearly, it's further along in developed countries than developing countries. But if you look at these data, where the developing countries are today is where the developed countries were 25 years ago. So it's consistent with the idea that it's the same phenomena, but they're lagging by 25 years. And in fact, if we look at just the numbers of children under 5 who were obese or overweight in the world in 2015, there were 9 million in developed countries, but 42 million in developing countries. Most of the problem is in developing countries. So this is truly a global epidemic. And today I'd like to discuss how the microbiome, what the microbiome might have to do with this. And to do this, I want to consider 3 different concepts initially. First is evolution. So this slide looks at evolutionary relationships of wild hominids. Here on the left side is the phylogeny based on host genes. Here's a recognizable hominid, wild hominid. And this has a particular branching pattern. There's a topology. And now on the right side, we look at the fecal microbiota. And this is work from Howard Ochman and his colleagues. And if you look at the branching pattern, you'll see that it's the same. So this congruence is strong evidence for vertical transmission of the microbiota. And it's also evidence that supports the idea of coevolution. And the time scale of this congruence here is about 8 million years. There's evidence that goes back even further than that, suggesting this shared evolution of microbes and other mammalian species. Now, the second concept I want to mention is about an age window. And this is a slide from the work of Tanya Yatsenenko and Jeff Gordon's lab. It includes work by Rob Knight and Maria Gloria Dominguez. They studied healthy people in the US, Amerindians, and Malawians. And here on the x-axis is age during childhood. Each circle is the composite of the gut microbiome in one individual. And the y-axis is called unifrac distance between children and adults. This is a measure of dissimilarity. And what the data clearly show is that when babies are born, their microbiome is very unadult-like, and then gradually and progressively it becomes more and more adult-like until it is. And what's remarkable to me about this study is that the age of transition is the age of 3 years. The first 3 years of life is when the microbiome is the most dynamic, when it's developing. It's also the period of time when babies are developing. They're developing their metabolism, their immunity, and their cognition. Third concept I want to introduce is a concept of equilibrium. This is work that I did with Denise Kirshner, a mathematician. It shows a schematic of the interactions between a co-evolved colonizing microbe and the host. And our idea of coevolution are microbes that know how to talk to the host and that receive conversation back. We've modeled this as an equilibrium. It's a dynamic equilibrium. Denise has shown that it's robust and resilient, can go on for decades. What I want to talk about today is what happens when these ancient relationships get perturbed. What happens when they're lost? So let's go back to transmission, to vertical transmission. We humans are mammals. We begin our life in a womb that is sterile or mostly sterile. Our first big exposure to the world of bacteria occurs when the water breaks. When the baby descends through the birth canal, it's covered by mom's microbes. The baby swallows the mom's microbes. There's skin-to-skin contact between mom and baby. The inoculated mouth inoculates the breast, and now microbes and breast milk substrate go in to become the founding members of the GI tract microbiome. There's oral transmission. This is the way it's always been for all mammals over these last 100 million years. Nature has a lot of redundant pathways. But now we're living in a modern world. And modern moms aren't the same as they used to be. They live in an environment with antisepsis, with antibiotics, with diets, with antibacterial substances. And babies aren't the same either. For example, babies may miss the passage through the birth canal. They may be born by cesarean section. In the United States, 32% of babies are born by C-section. In many countries of the world, the rates are 50%. So this is a big change in our ecology. Babies are bathed extensively. They get formula, which only superficially resembles human breast milk. It doesn't have the advantages of millions of years of coevolution and substrate. And of course, babies get lots of antibiotics. So on the basis of these ideas, over these last almost 2 decades, I've been thinking about something that I've called the disappearing microbiota hypothesis. And this hypothesis has 2 major tenets. First, that changed human ecology has altered transmission and maintenance of ancestral microbes, which affects the composition of the microbiota. We have evidence for that already. And especially important are microbes usually acquired early in life, since they affect a developmentally critical stage. Now, some years ago, Dr. Stan Falco and I enlarged this hypothesis, which is shown here, the effect of maternal status on the resident microbiota of the next generation. In our view, ancient moms had an ancient microbiota, and if they lost microbes before the next generation was born, and they couldn't get them back horizontally, then the next generation would be born at a deficit, and so on, and so on. And this is our view of what has been going on with the human microbiome over the course of the 20th century into the 21st century, that the changes in the microbiome have been cumulative across generations. Recently, I've expanded this work as well. Here's this step down that I showed before. This might be a country like the United States. This might be a country like India or China that began its development later, but is progressing very rapidly. These might be Amerindians in South America who haven't had much modernization. So the start of the loss and the pace of the loss may differ locally. Well, this is hypothesis, but there is already data that support the hypothesis. Here we're going to look at the disappearance of Helicobacter pylori, the major taxa in the human stomach, in 3 generations of Japanese families: grandmothers, mothers, and children. And we can see the kind of step we envisioned. Here we're going to look at the diversity in the fecal specimens in 4 human groups. This is work led by Maria Gloria Dominguez. We're going to look at diversity in U.S. children, children in Malawi, Guahibo tribal children, and a group of uncontacted Yanomami Amerindians on their first contact with Western people and Western medicine. And here on the y-axis is a metric of phylogenetic diversity. Here are the Yanomami, here's the US. These 2 groups are intermediate. We can almost imagine the steps here. And comparing the diversity in the Yanomami to the US is consistent with the hypothesis that we have already lost 50% of our diversity. This isn't the only study. There are several other studies pointing in the same direction. Here's a recent paper looking at 2 tribal groups in Africa. Here's the diversity in those groups compared to the US. Again, consistent with this big loss of diversity in developed countries. Now the question is, if this is true, what could be causing this? What's of sufficient magnitude to cause such a widespread change? And I'm going to raise the hypothesis that one of the important actors here is antibiotics. Antibiotics were one of the great discoveries of the 20th century. They've saved countless numbers of lives. And as a result, medical practitioners and the public have used antibiotics more and more and more. How much more? WHO estimates in a recent year that there were 73 billion antibiotic doses yearly worldwide. That's 10 doses of antibiotics for every man, woman, and child on Earth, and the numbers are increasing. In the United States, in 2010, the CDC counted 258 million courses of antibiotics. That's 833 courses per 1,000 population. 5 courses for every 6 people. This has been going on year after year. Extrapolating from the CDC data, children are getting nearly 3 courses of antibiotics by the time they're 2 on average, and by the time they're 10, about 10 courses of antibiotics. Pregnant women in the United States, more than 50% are receiving antibiotics sometime during pregnancy. This is just before the intergenerational handoff of the microbiota. So it's happening on a great scale, and in some countries, like China, for example, it's happening on an even greater scale. Now, the CDC looked at the geography of antibiotic use. The national rate is 833 per 1,000, Northeast 830, Midwest 868, about the national average, West 638. Here's state of Washington, nice and light in color. Here's the South, 936. Between the South and the West, a 50% difference in the per capita rate of antibiotic prescriptions. And there is no 50% difference in the rate of serious bacterial infections. This reflects differences in practice and culture. And just about every survey of antibiotic use shows enormous diversity and variation in usage. Now, especially interesting to me is to compare these 2 CDC maps from 2010, the geography of obesity on the left and the geography of antibiotic use on the right. These are observational data, but if you look at them, you can see that the patterns align pretty closely. They don't tell us anything about causation. And there have been epidemiologic studies that have asked the question, is exposure to antibiotics early in life associated with weight gain? This slide is from a study we performed with colleagues in the ALSPAC cohort of more than 10,000 children born in England in the early 1990s. And we asked about antibiotic exposures. The bottom line from this study is that children who were exposed to antibiotics during the first 6 months of life had increased adiposity when we controlled for other confounders. Since 2011, there have now been 16 studies that we could find that have asked the question about antibiotic use and adiposity. And nearly all of them show a positive relationship. There are a couple that don't show it, but if you look at the data carefully, I think you see it as well. But nevertheless, these are all observational data. Are there experiments that help us understand the relationship? Well, in fact, farmers have been conducting an experiment for the last 70 years. They learned that if they give low doses of antibiotics to their livestock, it promotes their growth. This growth promotion uses subtherapeutic antibiotic concentrations, something that we abbreviate as STAT. The farmers have found that it works from chickens to cows. That is a wide swath of vertebrate evolution. And the reason they use it so much is because it works. They have shown that virtually any antibiotic we use, regardless of its chemical structure, its class, its target, or its spectrum, works as long as it's an antibacterial. Antifungals don't work, antivirals don't work. Importantly, the earlier in life that the antibiotic is started, the bigger the effect on growth rate, the bigger the effect on feed efficiency. Feed efficiency is the conversion of food calories into body mass. This is what farmers are trying to do. So when we saw these data, it immediately suggested that we were looking at a developmental phenomenon. So we began studies in mice where we would give mice antibiotics or not. We would examine properties in the mice, look at the microbiome, look for relationships. In my limited time, I'm going to show you the key features of 8 experiments that we've done to address these questions. The first experiment was started by Dr. Il-Sung Cho when he was a fellow in my lab. Here we're looking at the adiposity of mice given antibiotics. Il-Sung used 4 different antibiotic regimens at the midpoint of the FDA-approved level on the farm, or no antibiotics. Here we're looking at percent body fat. We can see these illustrations. He found that giving the mice antibiotics increased body fat. This was our first evidence that antibiotics would affect body composition. And Il-Sung went on to show many other things, but I'm going to move ahead to the next experiment that was done by Laurie Cox when she was a graduate student in the lab. Laurie did an experiment that we call FAT-STAT. She asked, what are the combined effects of a high-fat diet and the STAT antibiotics on body composition? So she gave mice low-dose penicillin in their drinking water. All the mice were on normal chow, and at week 17 of life, half the mice were put on a high-fat diet. Here are the main results in terms of morphometry. Male mice and female mice, total mass, fat mass, and lean mass. So if we look at the male mice total mass, this black line is the control group. Normal chow, no antibiotics. Normal chow with antibiotics, they're bigger, just like on the farm. High-fat diet, bigger still. High-fat plus antibiotics, biggest. Lean mass increased on antibiotics, just like on the farm. Fat mass, big increase on high-fat diet, even more high-fat plus antibiotics. Female mice, many of the same trends. If we look at fat mass, the average female mouse on the high-fat diet gained 5 grams of body fat. High-fat plus antibiotics, 10 grams. They doubled the amount of body fat. The antibiotic potentiated the effect of the high-fat diet. It also shows that life is unfair, bigger effect in the female mice than the male mice. Now, we continued this experiment because up to this point, the mice were getting antibiotics for their whole life. So Laurie did an experiment we called Durastat to ask, are the changes durable with limited antibiotic exposure? So in addition to mice that received no antibiotics or lifelong antibiotics, we had a group that only had 8 weeks of antibiotics or only had 4 weeks of antibiotics. Here are the results for morphometry, total mass, lean mass, and fat mass. The black line is the control group. What we found is that all 3 antibiotic regimens were sufficient to increase total, lean, and fat mass. So 4 weeks early in life was sufficient for the full effect. We are interested in what's going on in the intestine, and we have many studies of this. I want to just show you one key study. Here's work by Jackie Leung and Peng Lok's lab. We're looking at the effects of STAT on intestinal Th17 populations. Here by flow cytometry, looking at the small intestine and the large intestine. However we look at it, Th17 populations are down in the ileal wall in the mice that receive the antibiotics. We'll come back to that. Now, what about the microbiome? So here we're going to look at the fecal community structure in mice at 3 weeks of age. And at 3 weeks, we essentially have two groups, a control group, no antibiotics, or all three antibiotic groups are still receiving the antibiotics at 3 weeks. Here's the principal coordinates analysis. Each circle is the composite of the gut microbiome in one individual. Black dots are control, oranges are stat. They're similar but not identical. That's not surprising. One group is receiving antibiotics, the other is not. Now we're going to go to 8 weeks. And at 8 weeks, we have 3 groups of mice. Control mice, no antibiotics. 2 groups still receiving antibiotics. And a 3rd group that got 4 weeks of antibiotics and then stopped. So now when we look at the principal coordinates analysis, the controls are in black. The stat are in orange. They're more separated than before, which is not surprising because one group is continuing to get antibiotics. But the group that got antibiotics for 4 weeks and then stopped, their microbiota have reverted to the normal. So the effect of the antibiotics on their microbiota was transient, but the effect on the phenotype was permanent. So this slide provides evidence that altering the gut microbiota during a critical early period in development has long-term consequences. Now, to continue this study, Laurie did a transfer of microbes from conventional mice to germ-free mice. We had mice that received low-dose penicillin or not. We sacrificed these mice. We harvested their cecal contents, and we gave them to germ-free recipients. And we followed them for 5 weeks. These mice did not receive any antibiotics at all. And here we're looking at morphometry, total lean and fat mass. Black line is the control group. What we found is that the antibiotic perturbed microbiota increased total mass in the recipients, no change in lean, increase in fat mass. So the altered microbiota were sufficient to confer the signal. How about immunologic phenomena? Here we're going to look at expression of genes involved in intestinal defenses. Here, we're going to start to look at the donor mice, Th17-related genes, antimicrobial peptides downstream of Th17s, transcription factors, cytokines. For all of them, we see that these populations of genes are down, as we've come to expect in the donor mice that are treated with antibiotics. But how about if we transfer the microbiota from these mice to the recipients? And in fact, we see the same phenomena as well. So this provides evidence that the immunologic signal is transferable with the microbiota as well. Now, up to this point, I've shown you work using the farm model, the STAT model, low doses of antibiotics every day in their drinking water. But human children are getting pulses of antibiotics. They're getting courses of antibiotics to treat their ear infections and their throat infections. So Yael Noble, a student in the lab, tested the hypothesis that a series of short therapeutic dose courses of antibiotics administered early in life will sufficiently change the gut microbiome to alter host phenotypes. Here's the design of Yael's study. She looked at 2 different antibiotics that represent commonly used agents, amoxicillin, the most widely used antibiotic in childhood. Those of you who are parents will recognize that. Or tylosin, a macrolide. This might be the Z-Pak for mice, or a mixture of the 2. Mice got 3 courses of antibiotics. The first came through their mom's milk. Then 2 were in chow. Then we put all the mice on a high-fat diet to bring out the phenotype. I want to show you just a couple of key results from this study. First, we're going to look at what happens to the intestinal populations, looking at the alpha diversity, the richness and the evenness. The blue line is the control group. What we see is that the amoxicillin has some effect, and even 100 days after the antibiotic pulses, the populations have not recovered. The macrolide has even a more profound effect, also no recovery. We see this in both richness and evenness. So in contrast to the subtherapeutic antibiotics, the therapeutic antibiotics have extended effects on the developing animal microbiota. Now, Alex Alexienko did a cluster analysis using Kolinsky analysis. He found that 4 clusters were best described. We could call these the maternal cluster in green, the high-fat cluster in red, and 2 different antibiotic clusters in blue and yellow. And here's, if we look at the mice in the experiment, over the course of the experiment, by group, we can see several patterns. So in the controls, they begin life with their microbiota in the mom cluster. Then when the high-fat diet begins, there's a strong shift. The strong selection of the high-fat diet changes the character of the microbiota. When they're on amoxicillin, there's some perturbation, but it's not too extensive. But if they're on the macrolide or the combination with the macrolide, there's a huge shift in the microbiota. And finally, the strong selection from the high-fat diet brings them to the new normal. So we can see that there can be very big effects on the microbiota of early life antibiotics, and that all antibiotics are not equal. And a recent study of human children in Finland pointed this out as well. Now, the final problem I want to talk about today is about type 1 diabetes. Type 1 diabetes is an autoimmune disease that is doubling in the world every 20 or 25 years, or at least in the developed world where it can be measured. So the question is, why is this autoimmune disease going up so much? Alexandra Livanos, an MD PhD student in the lab, had the hypothesis that antibiotics are promoting type 1 diabetes. And she tested this hypothesis in NOD mice. These are mice that spontaneously get an autoimmune form of insulitis. Her hypothesis is that early life antibiotics would change the gut microbiome, they'd change intestinal gene expression, they would change T cell subtypes, and this would lead to increased type 1 diabetes. Ali has now graduated. She's shown every point of this, and I want to illustrate a couple of the key points. So first, we're going to look at type 1 incidence in the NOD mice. Female mice and male mice, in NODs, they differ in their rates of diabetes. Females get more. The black line is the control group. In this experiment, female mice had no antibiotic phenotype. However, the male mice receiving the PAT, or the pulsed antibiotic, therapeutic dose antibiotics, had acceleration of diabetes and enhancement, and we've now confirmed this 3 times. From this experiment, we also had subgroups of mice who we sacrificed at 6 weeks. This is before the development of diabetes. So we wanted to see what's going on in their tissues beforehand. So first we looked at the pancreas, looking for insulitis in 6-week-old NOD mice. Looking at female mice and male mice, control, and the 2 antibiotic groups, we see the antibiotics increase insulitis, and this difference is significant in the male mice that show the strong phenotype. How about the intestinal wall? Now we're looking at the effects of antibiotics on T regulatory cells in the small intestinal lamina propria. Here are the female mice, no effects on these cells, consistent with no phenotype. Male mice, higher levels in the controls, and this is consistent with them having less diabetes than the females. With antibiotics, they become more female-like. How about Th17 cells in the lamina propria? Very similar phenomena. No change in females, higher levels in the males brought down by the antibiotics. What about the microbiota? This is a network analysis of the co-association of particular OTUs in fecal samples from 6-week-old male control and PAT mice. This was done by Dan Knights at the University of Minnesota. In this figure, each black dot is an OTU, each circle is a sample. PAT is in red, control is in blue. We can summarize the main findings by saying that there's a decreased number of OTUs in PAT compared to control. There are very few shared OTUs between control and PAT. And this work has enabled us to identify particular taxa that are significantly underrepresented in PAT, which become candidates for further study. Now, the next question we had is this an antibiotic effect or is this a microbiota effect? So Shusong Zhang did an experiment where he gave PAT mice a single course of antibiotics between day 10 and 15 of life. And 2 days later, he sacrificed the mice who got antibiotics or not and gave those contents to germ-free mice. In this case, we gave it to C57 black 6 mice because that was all that we had available. But we still could look at T regulatory cells and Th17 cells in the spleen and the lamina propria. And what we found is that the microbiota from the PAT-treated mice had lower levels of these, just like we saw in the original NOD mice. And this study provides evidence that the microbiota in this experiment as well is capable of conferring an immunologic signal. Finally, Shusong has done a recent experiment where, in addition to our regimen of 3 pulses of antibiotics, he's asked what happens if he just uses a single pulse of antibiotics, a single course early in life, between days 5 and 10 of mouse life. And here are the results of the study. Here's the Kaplan-Meier analysis in male mice and female mice. In blue are the 2 control groups. In red are the 3 PAT course or the 1 PAT course. So even a single course changed the incidence of diabetes in these mice. In female mice, we see the effect is variable, as we saw before. With 3 PAT, we see some effect. With 1 PAT, we do not see an effect. So we're continuing this work, and in fact, Shusong presented this yesterday at the Endocrine Society meetings. So how do we put all this together? Our hypothesis is that antibiotics are having an impact on long-term physiology through changes in the microbiota. Our idea is that in health, we have co-evolved with a gut microbiota, which is having stereotypic conserved conversations with stem cell populations and stem-like populations. And these conversations are guiding early life cellular development, if they're mesenchymal cells, adipose, muscle, bone, or immunocytes, and this is leading to long-term physiology. In contrast, a perturbed microbiota by antibiotics or Csection or compounded factors leads to an abnormal microbiota having abnormal conversations, leading to abnormal early life development and long-term physiologic consequences. So some slides ago, I presented this idea of how we're stepping down in the microbiota. And when I wrote this in 2016, last year, now the question is, what's the future going to bring? Are we going to have further decline? Are we going to arrest the decline? Are we going to reverse the decline through restorative steps? So here's my view of medicine of the future. New analyses of child health. The pediatricians of the future will examine babies, and they'll examine babies' diapers. And they'll ask, do these babies have all the global microbes that babies should have? Do they have all the personal microbes that this baby of this genotype and other biomarkers should have? And if not, the doctors of the future are going to reach for those microbes from the shelf, that will be part of the pharmacopoeia, and administer them to these babies. And ultimately assess, did it work? Have they optimized their phenotypes for long-term health? I want to conclude by thanking the many people who've done the work I've talked about. I've recognized many of them as we've gone along. Many others worked on other aspects of the projects and with lots of support from different government and foundations. And finally, a shameless plug of my book, Missing Microbes. You can see that everyone is reading it. Thank you very much.
We can take a few questions.
So are kids who have pets early in life, are they less obese than those who don't?
Is your question pets?
Pets, yes.
So there's a little data about it that suggests that kids who have pets have a healthier microbiome than others. I'm not certain. I don't know. I'm agnostic about this. The one thing I will say is that the microbes that dogs have evolved for dogs, and the microbes that cats have evolved for cats. Whether those microbes have an influence on our ancient human microbiota is uncertain.
Back there.
Hi, my name is Yoel Sadowski, and I'm from McGee Women's Research Institute in Pittsburgh. And I'm very interested in the very beginning of life and the points that you made about change of the microbiome during birth and the different effects that play a role there. In addition to C-section or no C-section and antibiotics or not, there are a number of other dominant changes that take place right at birth. The child is being exposed to mother's skin microbiome, as you mentioned, breast milk, father's involvement, and of course the maternal GI microbiome. So these changes take place in the very first few days of life. In addition to the association that you showed, do you really have data to show that C-section or not, or antibiotics or not, truly change the child's microbiome 2 or 3 weeks later? In fact, there's some recent data that show that maybe it doesn't. And this is important for people because there are some mothers who undergo C-section who are asking now to have a sponge placed in the vagina and reinstating the vaginal microbiome. What is really the science behind it?
Yeah. So this is a wonderful and important point that you raised. And there are many points that you raised. So let me start maybe at the beginning. The first is to say that there's data from starting 5 years ago from a PNAS paper showing that the microbiota in kids born by C-section is different from the kids born vaginally. That's been confirmed several times. The recent paper showed that the microbiota varies by body site, which is not surprising, that the microbiota that evolves on the arm is different than the microbiota in the mouth. But if you look at it carefully, you will see the same C-section vaginal signal as well. We had a paper last year in Science Translational Medicine, and there was a paired paper with it. All of these showed effects of C-section on alpha diversity, beta diversity, and particular composition and maturation of the microbiota. We also showed effects of formula feeding and effects of antibiotics. Unfortunately, we are impacting the early life microbiota during a period of time when it has very low resilience, the period of time when it probably has the least resilience, with major impacts such as maternal antibiotics, cesarean section, extensive washing. I thought you might mention that, where babies are being washed with chlorhexidine now routinely, formula, which doesn't have the human milk oligosaccharides, and of course, early life antibiotics. And recent work by Maria Gloria Dominguez showed that kids born by C-section have abnormalities, and that study went out to a month. Our work shows it goes out beyond a year. And with the sponge, she can at least partially restore the microbiota. And this is consistent with the idea that we're going to have to restore microbiota. So there are certain professional groups that think that vaginal birth is risky, and it has some risks, but it seems to have many benefits, and evolution has taught us that as well.
I should point out we have a panel discussion at the end of the 3 speakers, but we will take one question now.
Question for you. To what extent can these changes be reversed? If you do fecal transplants, are the changes reversible, and are they sensitive to what age you do it? Can the gut microbiome be stabilized through these things?
Yeah. So I mentioned the study of the vaginal restoration, and that's work that is ongoing. Our work on restoration is entirely limited to mice. And I can tell you that some of our preliminary work shows that we can get at least some partial restoration. But our work suggests that there is really a window, and that when the window is passed, the restoration is going to be of less and less efficacy. So to us, we're really interested in understanding the very early life changes, and so that we can find the best restorative strategies. This is work that's in progress in our lab and several labs around the world.
Okay, so as they're setting up, I'll ask you one last question maybe, but they need to set up, but since I have a few minutes. So in terms of restorative interventions, do you see there's going to be some type of matching of microbiomes between donors and the people who are receiving the microbiomes, and what kind of that matching would that be?
So it's fair to say that we are at the earliest stages of understanding the human microbiome. There are many studies that are looking at adults who have inherited certain microbes and then they've changed over the course of 40, 50 years of human life. Again, we're focusing really on the earliest life, on the ones that are inherited. And most of the work that's going on now is looking, as you said, is looking at taxa, but we have to look at functional groups. We have to look at strain differences because all E. coli are not the same and all bifidobacteria are not the same. And so it's an early science. I think that it will start reaching fruit 10 or 20 years from now, that it will really be in the realm of clinical application, where we're going to have enough specificity to answer your question.
Thank you, Marty.
Thank you. Thank you, guys.